Scientists have used a device that fits in the palm of the hand to sequence the human genome. They say the feat, detailed in the journal Nature Biotechnology, opens up exciting possibilities for using genetics in routine medicine. It is a far cry from the effort to sequence the first human genome, which started in 1990. The Human Genome Project took 13 years, laboratories around the world, and hundreds of millions of dollars. Since then there has been a revolution in cracking the code of life. Professor Nicholas Lohmann, one of the researchers and from the University of Birmingham, UK, told the Newsbay, "We've gone from a situation where you can only do genome sequencing for a huge amount of money in well-equipped labs to one where we can have genome sequencing literally in your pocket just like a mobile phone. That gives us a really exciting opportunity to start having genome sequencing as a routine tool, perhaps something people can do in their own home." Sequencing technology has the potential to change the way we do medicine. Analyzing the mutated DNA of cancers could be used to pick the best treatment. Or inspecting the genetic code of bacteria could spot antibiotic resistance early. Professor Lohmann used the handheld device to track the spread of Ebola during the outbreak in West Africa. Many companies are racing to sequence DNA faster and cheaper. The handheld device used in the study was developed by the company Oxford Nanopore. The technology works by passing long strands of DNA through a tiny hole, the eponymous nanopore. The building blocks of DNA are 4 bases known by their letters A, C, G and T. As each passes through the pore, it creates a unique electrical signal that allows researchers to determine the DNA sequence. The research group say the approach was around 99.5% accurate, but also allowed them to look at parts of human DNA that had been poorly studied. The commonly used short-read method of sequencing DNA involved breaking it up into short fragments, sequencing those and piecing it all back together like a jigsaw. But the challenge is that some fragments of the genetic code look incredibly similar, so it becomes a near impossible puzzle made of identical pieces. Professor Matthew Luce, from the University of Nottingham, UK, said the nanopore tech analyzed longer strands of DNA so we can read parts of the genome not seen before. This includes the tips of chromosomes, called telomeres. Professor Luce told the Newsbay, "Telomere length can be quite important in cancer and aging. It's difficult in short reads because of the repeats, but we can see and start to map those things." But while the cost of the sequencing is tumbling, there remains a big barrier. Being able to rapidly read the genetic code is not the same as understanding what it says. Dr. Sobia Raza, the head of science at the PHG Foundation Genomics think tank, told the Newsbay, "Our ability to sequence whole genomes quickly and cheaply continues to improve. But short-term patient benefits also depend on how well and how fast we can analyze and make sense of the genomic data, and that is still quite a challenge."